Welcome to this Explainer Masterclass. Today, we're diving deep into the microbial world's secret weapon for evolution, genetic exchange. Now, this is not your standard parent-to-child inheritance. No, this is something far more dynamic. We're talking about bacteria sharing genetic blueprints with their neighbors, sometimes not even the same species. It's this ability that allows them to create superbugs and adapt to new environments in what feels like the blink of an eye. We're going to dissect the three core ways they do this, a process called horizontal gene transfer, to really understand how it shapes the world all around us. So here's our roadmap for today. We'll kick things off by defining horizontal gene transfer, or HGT. Then we'll get into the nitty-gritty of the three main pathways. First, transformation. That's bacteria picking up stray DNA from their surroundings. Then, transduction, where they use viruses as little genetic messengers. And finally, conjugation, which involves direct cell-to-cell contact. After that, we'll see how this new DNA actually gets integrated through recombination. And to wrap it all up, we'll put all three mechanisms side by side to really lock in those key differences. All right, Section 1, an introduction to HGT. The best way to think about this is like a microbial information superhighway. You see, while complex organisms like us pass genes down vertically from parents to offspring, prokaryotes have this incredible ability to pass genetic material horizontally to completely unrelated individuals right next door. It's not inheritance in the classic sense, it's sharing. And this process, right here, is the primary engine that drives bacterial evolution and gives them their mind-boggling speed of adaptation. Now, this genetic sharing happens in three main ways. First up, we have transformation, where a bacterium basically scavenges a free-floating piece of DNA directly from its environment. Second is transduction, which is a bit more elaborate. It uses viruses, specifically bacteriophages, as these tiny genetic delivery trucks. And third, there's conjugation, which is the closest thing bacteria have to mating, where two cells physically connect to exchange DNA. So let's dive in and explore each one of these in detail. Okay, let's get into our first pillar here in Section 2, transformation. This is all about taking up DNA directly from the environment. Just picture it. A bacterium dies and bursts open. It lyses. Its chromosome breaks apart into little fragments that are now just floating around. Well, another bacterium can come along, find one of these fragments, and absorb it, potentially gaining brand new genetic traits in the process. So, the formal definition is that transformation is the genetic alteration of a cell from the direct uptake of foreign or exogenous DNA. The absolute key here is that this DNA is naked. It's not packaged inside another cell or a virus. It's just a raw piece of genetic code out there in the environment, waiting for a recipient. And what's really amazing is that this was the very first mechanism of bacterial gene exchange ever discovered. Now, not just any bacterium can do this. It has to be in a very special physiological state called competence. And there are two ways to get there. Some bacteria, like certain species of Streptococcus or Bacillus, have what's called natural competence. It's a built-in, genetically programmed ability they can just switch on, which allows them to bind and pull in DNA. But others, like the famous lab workhorse E. coli, they're not naturally competent. So in the lab, we have to induce competence artificially. We use chemical treatments or even an electrical shock, a process called electroporation, to poke temporary holes in their membranes so the DNA can get in. So how does this natural transformation actually happen? Well, it's basically a three-step process. First, a piece of double-stranded DNA just bumps into and binds to the surface of a competent cell. Simple enough. But then the second step is fascinating. The cell starts to pull the DNA inside, but as it does, it chews up and degrades one of the two strands, so only a single strand of that DNA actually enters the cell. And finally, for this new code to actually be passed on to future generations, it has to be woven into the host's own chromosome. This happens through a process called homologous recombination, which we're definitely going to circle back to later. All right, let's move on to our second mechanism in Section 3, transduction. So if transformation is like finding a message in a bottle, transduction is like getting a package delivered by a special courier. And in this case, the couriers are viruses, specifically viruses that have evolved to infect bacteria. These bacterial viruses are called bacteriophages, or just phages for short. Now, these aren't just passive delivery vehicles. They are incredible biological machines that are purpose-built to inject their own genetic material into bacteria. To really get a handle on transduction, we first have to understand the two main life cycles of a phage, because it's during these cycles that the transfer of bacterial DNA happens, usually completely by accident. First up is the lytic cycle. This is a brutal, hostile takeover. Let's follow along here.
First, the phage latches on and injects its genome. Almost immediately, the host's own chromosome gets degraded, chopped into pieces. Then, the viral genes completely hijack the cell's machinery, forcing it to make copies of the viral genome and build new viral proteins. These parts then get assembled into hundreds of new phages. And finally, the cell gets so full it just bursts, or lyses, releasing all those new viruses to infect other cells. As we'll see, mistakes made during this aggressive cycle are the key to one type of gene transfer.
The other path is the much stealthier lysogenic cycle. Here, the phage DNA doesn't immediately take over. Instead, as we can see, it cleverly integrates itself right into the host's chromosome. This dormant piece of viral DNA is now called a prophage, and the bacterium carrying it is called a lysogene. The prophage just lies low, getting a free ride and being replicated every single time the bacterium divides. This can go on for generations and generations, until some kind of stress, like UV light, triggers the prophage to cut itself out of the chromosome and kickstart that destructive lytic cycle.
Okay, so here's where the gene transfer comes in. Generalized transduction is basically a mistake that happens during the lytic cycle. Remember how the host chromosome gets all chopped up? Well, during the assembly stage, every once in a while, the phage's packaging machinery accidentally grabs a random fragment of that bacterial DNA and stuffs it into a new phage head or capsid instead of its own viral DNA. This creates what we call a transducing particle. It looks just like a normal phage on the outside, but it's carrying bacterial genes. When this particle goes and injects its contents into a new bacterium, it's actually transferring genes from the previous host. The critical thing to remember here is that any part of the bacterial genome can be transferred this way. It's completely random.
Specialized transduction, on the other hand, is an error that happens during the lysogenic cycle. It occurs when the prophage is triggered to leave the host chromosome. Sometimes, this cutting out process, or excision, is a bit sloppy. The phage DNA loops out incorrectly and ends up taking a few adjacent bacterial genes along with it, while accidentally leaving some of its own viral genes behind. The result is a defective phage that carries a very specific, not random, set of bacterial genes. Which ones? Only the ones that were located right next to where it was integrated on the chromosome.
And this slide shows that sloppy excision perfectly. You can see the prophage integrated in the chromosome. It's supposed to loop out nice and clean. But sometimes, as you can see here, it makes a mistake. It loops out incorrectly, grabbing a piece of the neighboring bacterial DNA while leaving some of its own DNA behind. This hybrid DNA then gets packaged into new phages, creating these specialized transducing particles that can deliver a very specific set of genes to the next cell they infect. This brings us to section 4, our third and final pillar of HGT, conjugation. Now, this is the most complex of the three mechanisms we're looking at, and it's defined by one absolute key requirement: direct physical contact between the donor cell and the recipient cell. The discovery of conjugation is a really classic story in microbiology. Back in 1946, Lederberg and Tatum were working with two different strains of E. coli, which we can see here as strain A and strain B. Now, both of these strains were what's called auxotrophic. They each had genetic defects that meant they couldn't produce certain essential nutrients on their own. And you can see, when they were plated alone on a minimal medium that lacked those nutrients, neither could grow. But, and here's the kicker, when they mixed the two strains together and then plated that mixture, colonies appeared. These new cells were prototrophs. They had somehow regained the ability to grow. The only possible explanation was that the two strains had exchanged genetic material. But how did that exchange happen? Could it have just been transformation? To figure this out, Bernard Davis designed this absolutely elegant YouTube experiment. He put the two auxotrophic strains in opposite arms of the tube, and he separated them with a filter. Now, this filter let all the liquid, nutrients, and any potential naked DNA pass through, but the pores were too fine for the bacteria themselves to cross. And the result? No growth. No prototrophs formed. This was the proof. It proved that transformation wasn't the answer, and that direct physical contact between the cells was absolutely required for this type of gene exchange. So what's mediating this physical contact? Well, the key player here is a special kind of plasmid called the F plasmid, or the fertility factor. A plasmid is just a small circular piece of DNA that exists separately from the main chromosome. The F plasmid is a specific type called an episome, which means it has the ability to either exist on its own or integrate itself into the bacterial chromosome. Most importantly, it contains all the genes needed for conjugation, including the genes to build a structure called a sex pilus. A cell that has this F plasmid is called F+, or F+, and it can act as a donor. A cell that doesn't have it is called F-, and it acts as the recipient. Okay, let's walk through this process. An F plus donor cell extends its sex pilus, latches onto an F recipient, and pulls the two cells together. A bridge forms between them. Now for the crucial part. The F plasmid in the donor is nicked. One strand is cut, and a process called rolling circle replication kicks off. As you can see in the diagram, one strand of the plasmid is peeled away and transferred into the recipient cell. And as this is happening, both cells are simultaneously synthesizing the complementary strand. So the donor keeps a copy, and the recipient builds its own copy. What's the end result? The F cell becomes an F plus cell, and the original donor stays F plus. It's an incredibly efficient way to spread the plasmid itself through a population. But what happens when that F plasmid, being an episome, actually integrates itself into the main bacterial chromosome? Well, this creates a really special type of donor cell called an HFR strain. HFR stands for high-frequency recombination. Now, it's still a donor cell because it has all the genes for conjugation, but now those genes are part of the chromosome itself. And these HFR cells get their name because they transfer chromosomal genes to recipient cells at a very, very high rate.
So what happens when an HFR cell decides to mate? The process starts the same way, but look what gets transferred. The integrated F factor starts the replication process, but now it starts to drag the entire host chromosome along with it. It's kind of like trying to photocopy a whole book by starting on a page somewhere in the middle. This transfer is really slow. It takes about 100 minutes for the whole E. coli chromosome, and that fragile connection between the cells almost always breaks before the full transfer is finished. So here's the key result. The recipient gets a big chunk of new chromosomal genes, which is great for recombination, but since the very tail end of the F factor almost never makes it across, the recipient itself usually stays F negative.
And there's one more variation on this theme. Just like an F plasmid can integrate to form an HFR strain, it can also cut itself back out to become an independent plasmid again. And, just like we saw with specialized transduction, this excision process can be imprecise. If the F factor loops out incorrectly and snags some of the adjacent chromosomal DNA with it, it forms what we call an F prime, or F prime plasmid. Now, when an F prime cell conjugates, the process is super efficient. It transfers the entire F prime plasmid, including those specific chromosomal genes it's carrying. So the recipient is converted into an F prime cell. But here's the really fascinating part. The recipient cell now has two copies of those specific bacterial genes, one on its own chromosome and one on the newly acquired plasmid. This creates what's known as a merazogote, a cell that is partially diploid. This whole process is an incredibly effective way to rapidly spread very specific, often useful, genes throughout a population. So far, we've really been focusing on the delivery mechanisms, all the clever ways bacteria get DNA from a donor to a recipient. But what happens next? Just getting the DNA into the cell isn't enough. For that new DNA to become a stable, heritable part of the recipient's genome, it generally has to be physically integrated into the chromosome. So let's look at this absolutely critical step in Section 5. The main way that transferred chromosomal DNA gets integrated is through a process called homologous recombination. And that word homologous is key here. It means that the process requires the incoming DNA to have a sequence that's very, very similar to a region that already exists on the recipient's chromosome. The cell's own machinery, driven by key proteins like one called Rec-A, sees this similarity, lines up the two DNA molecules, and then facilitates a physical swapping of the DNA strands. A piece of the recipient's original DNA is literally cut out and replaced by the new donor DNA. This is how new versions of genes, or new alleles, are stably incorporated, creating a brand new recombinant cell. And this brings us to the absolutely immense significance of horizontal gene transfer. I mean, this isn't just some interesting quirk of microbiology. This is the central force driving bacterial evolution. This is how bacteria adapt so incredibly quickly. And in a clinical context, this mechanism is directly responsible for one of our biggest public health crises, the rapid spread of antibiotic resistance. A single bacterium can pick up a resistance gene on a plasmid through conjugation and then rapidly share that plasmid with many other bacteria, even bacteria of different species, creating a population of superbugs in a terrifyingly short amount of time. Okay, we have covered a lot of complex ground. So for our final section, let's pull everything together and consolidate what we've learned with a direct, side-by-side comparison of the three mechanisms. This should really help clarify the key distinctions and make them stick. This table really sums it all up beautifully. Let's just walk through it. First, look at the DNA source. For transformation, it's free DNA from lysed cells. For transduction,
the DNA is packaged in a bacteriophage, and for conjugation, it comes from a living donor cell. The need for physical cell contact is a huge differentiator. It's a no for both transformation and transduction, but an absolute yes for conjugation. And this leads us to the final point, sensitivity to DNase, which is an enzyme that degrades DNA. Because transformation uses exposed, naked DNA, it's sensitive to this enzyme. But in transduction and conjugation, the DNA is protected, either inside a virus or inside a cellular bridge, so they are not sensitive. These are the classic diagnostic features that let us tell these three mechanisms apart.
So we'll end with this thought. We've been exploring an ancient genetic network, a system that has allowed microbes to adapt and thrive for billions of years. It's precisely what makes them so resilient. But it is also the very same system that drives the spread of antibiotic resistance, which is one of the greatest threats to modern medicine today. The challenge for us, and also the opportunity, is whether we can use our deep understanding of this network to actually fight back. Could we intercept these genetic messages? Could we turn the system against itself? The answer to that question is really going to shape the future of medicine. Thank you for joining us on The Explainer.